We have a lot of speakers today in various areas. We are having a future forward-looking day, and there's no better way to start that off than with my colleague, Dr. Peter Newman, who is world famous in the platelet arena. And Peter and I have known each other for longer than either of us would care to count up. And Peter has had a lot of impact on platelet biology field. He's still extremely active. He's very well published, but he's also one of those people who's very actively involved in more than just the research in his own laboratory. So Peter has an important administrative role. He's the Vice President of Research at the Blood Center of Wisconsin, which is one of the very research-intensive blood operators in the U.S. He's a professor of both cell biology and pharmacology at the Medical College of Wisconsin. He also has made a lot of contributions to the professional societies to which many of us belong. So he's been very active in the sort of senior ranks and the organizations of all of the American Society for Hematology, American Heart Association, and the International Society on Thrombosis and Hemostasis. So for those of you who have been involved in any of these societies, you know that just being involved with one takes a huge amount of time and effort. So kudos to Peter for being so generous to the profession to put his time into those societies. So Peter is going to talk to us today about some very exciting new things in the platelet field. And first of all, Ed is going to come up here and fix the slides because I don't know how to work his computer.
Thanks, Dana. You're welcome. Good job. Okay, Peter, you can come up too because I'm not giving you a talk. I can't give you a talk. Where are you? There we go. All right. I'm asking all speakers, please just use the cursor. It's a huge cursor. Okay? That way you will face forward. It's all yours.
Thanks, Ed. Well, good morning, everybody. Thanks for getting up early. Not too early for me because it's 10 o'clock in Milwaukee. This is Milwaukee during our 4 days of summer. The lake melts and the boats come into the water, and the leaves don't fall off the trees for another couple weeks. The place I work is called the Blood Research Institute, and it's a two-story, it's about 80,000 square feet, so that's about 8,000 square meters building, and it's solely dedicated to blood and blood research. We have about 27 basic science investigators and 9 clinical investigators. It was opened in 1991 and added on to in 2007. And the vision for this whole thing is due to a guy who's very famous in transfusion medicine named Dick Astor, who turns 87 this summer and still comes in on Saturdays. And sends more abstracts to ASH every year than I do. He won't let me catch up to him. So what I'm going to talk to you about today is the production of platelets from iPS cells and trying to make custom platelets. And the other thing I'm going to do is talk to you about a mouse model that we're developing for this disease that I'm going to tell you about that some of you might know about. So I'm sure in this audience everybody knows that platelets have a whole bunch of different receptors on their surface. Some of them are involved in their agonist receptors that are involved in activation. There are adhesion receptors that tether platelets to von Willebrand factor, which is immobilized on collagen. And then at the end of the day, there's an integrin called GP2B3A or alpha-2B beta-3 that becomes activated and binds a linker molecule, fibrinogen, which is very abundant in plasma. It's the third or fourth most abundant plasma protein at 3 or 4 mg per mil. And so that's the functional part of all these, and it's important to get it all to work. GP2B3A was crystallized about 15 years ago, and the beta subunit, or GP3A, is here in red. It has a ligand binding domain, and it has a stalk, and the membrane's down here. And I hope you can see these slides okay. I don't know if the lights in the front can come down just a bit. At least over the screen. And the alpha subunit, or GP2B, has a beta propeller region. And one of the cool things that this integrin does is when it gets activated by binding things to its cytoplasmic domain, it straightens up kind of like a jackknife, and this ligand binding domain here then is the thing that binds fibrinogen. So a very long time ago, there's an allelic form of GP3A, and this is the out of Africa slide, and we'll call it the PLA1 penase form of GP3A, and this is human migrations over the last 75,000 years. And some folks who left Africa got zapped and changed this to something called PLA2. And PLA2 left Europe about 500 years ago, and people of European descent in North America, about 15% of people carry this form, which was serologically defined in the early 1960s. So there were PLA1 positive and PLA1 negative or PLA2 individuals. Meanwhile, about 70,000 years ago, other humans left, and they got zapped in a different region of the gene and converted to the PEN-B serotype. And you can find this PEN-B serotype in Asia, and you can find it in Eskimos, and you can find it in Navajo Indians, and you can find it in the Mapocho Indians in Chile. So you can watch the Bering Strait happen just by following this antigen that's on platelets. And it turns out that if you have mismatched alleles for PLA1 or PEN-A and PEN-B, that it can result in a couple alloimmune disorders. Alloimmunity is the immune system reacting to a SNP on another human allele, as opposed to isoimmunity, which is a protein like you've never seen before, like often happens in factor VIII inhibitors. And one of the disorders is called post-transfusion purpura, and it's where a multiparous woman receives a blood transfusion and makes antibodies to an antigen that she hasn't seen before, and it clears those platelets and for some reason also clears her own, and it usually resolves within a couple weeks. It can be fatal, but it usually isn't, but it's scary. The one that I'm going to talk to you about now, which happens much more frequently, is called neonatal alloimmune thrombocytopenia, and it's due to maternal alloimmunization against fetal platelet antigens that are inherited from the father. So for example, if the mother is PLA2 and the father is PLA1, she can make a serological antibody to this form that she's never seen before. The babies are born thrombocytopenic, and through the trauma of birth, and can even happen in utero in the first pregnancy, they can have a cerebral hemorrhage, and then they're sort of institutionalized for life. So this happens as frequently as 1 in 1,000 live births, and it's the most frequent cause of severe isolated thrombocytopenia in maternity wards, and as I showed you on the last slide, its most destructive consequence is intracranial hemorrhage. It's also been linked to miscarriages, but that's not been very well studied. And the transfusion medicine folks call these things HPAs, or human platelet alloantigens. And the one that's most commonly implicated in the Caucasian population is HP1A, 1B, or PLA1, PLA2. But this PLA2 allele didn't make it to Asia, except for occasional sailors. So almost all Asians are this PLA1 type. And we found out a very long time ago that this is caused by a single SNP at a residue. It changes a leucine to a proline. And so this usually occurs, as I said, when an A1, A2, or an A1, A1 father, which is the higher frequency allele, 0.85, passes on this to a fetus that's being carried by a A2, A2, or a proline mother. And about 1 in 50 Caucasian mothers are PLA2 homozygous, so they're the ones who are at risk. And what's thought to happen is that these PLA1 positive platelets shed microparticles, they cross the placenta, immunize the mother, and then the maternal antibodies are passed back to the fetus where they clear the platelets from circulation. So this residue is located right in this little region right here, and I have a high-resolution picture of what this looks like. There's a little domain that has about 50 residues in it called the PSI domain, and this leucine-proline polymorphism is right at the tip of what looks to be a fairly mobile loop, and mobile loops are the things in immunology that are most likely to be immunogenic. And so you can imagine when there's a proline here, it would kink this differently, and so a proline mom sees this leucine sticking out and makes these anti-PLA1 or anti-leucine antibodies. And of course, this ancestral allele where I have leucine at 33 and PEN is an argtoglin at 143, this is what you can find in higher apes. But over the years, through evolution, there have been a lot of different SNPs that have formed in the human population. And so you can see some of the major alleles of GP3A right here, and so you can see the gene frequency of this PLA2 form or PLA1 form with the leucine 33 is about 0.85, and this is in Caucasians. It differs in New Guinea, it differs in Asia, it differs in Australia, just like ABO blood groups do. And the PLA2 allele, it has a 15% gene frequency in Caucasians, and you can see all these other SNPs. And these were all identified between about 1990 and 1996. There are still occasional ones of very low frequency antigens that are discovered. And well before HIPAA, they used to be given the names of the mother who found them. So she's gone now, but this is Mrs. Pena. She was a Navajo Indian, and et cetera. So, and some of these are quite rare, like this SR antigen is found only in the Black Forest in Germany. So, but, mutations happen, and the immune system detects them, and it's reported by severely thrombocytopenic infants. So it's the type of thing where the human disease points us right where we need to be looking. And you can see here's a GP2B3A, here's a schematic representation of these SNPs and where they occur on these proteins. It's not the only polymorphic protein. Alpha-2 beta-1, the collagen receptor, has some SNPs that this is probably the third or fourth most common cause of neonatal alloimmune thrombocytopenia here. But there are others as well. There's some on GP2B, et cetera. Some use carbohydrates that are nearby as part of the epitope, and many of them don't. So, and like I said, they're still being discovered with very low frequency, and you can see them pop up from time to time in transfusion in the journal. So there are some challenges in managing these things. Platelets expressing these rare alleles are not readily available because they're rare. So if you want to do typing for the antibodies, it's tough because you don't have target proteins in a lab. And as you know, multiparous women usually have HLA antibodies too against class 1 antigens. And so if you're doing whole cell assays, these class 1 antibodies can mask the presence of the platelet-specific antigens. And the third thing that I'm going to address in the last third of this talk is there's no way to prevent alloimmunization right now of women who are exposed to polymorphic platelet proteins. So what we decided to do is use CRISPR gene editing technology to modify induced pluripotent stem cells so that they can express these rare alloantigenic epitopes. And then turn them into megakaryocytes and platelets. And it would also be handy if they'd also fail to express class 1 so that the class 1 antibodies won't bind and mask the presence of these platelet-specific antigens. So then we grow them in culture, and we differentiate them into megakaryocyte progenitor cells and platelets, and then we call these things designer platelets. And we can use them for diagnostic purposes immediately, investigative purposes, and right now we don't make enough platelets to have therapy, but we're sort of at the beginning stages of this technology. So it's something to, I think, since this is supposed to be the future of transfusion medicine, this is probably farther into the future than I'll be dealing with, but some of the young people in the audience might be transfusing designer platelets. And then, like I said, in the last third, I want to talk to you about how we've made a transgenic mouse using CRISPR that expresses this human PLA1 antigenic epitope to look at some therapies that are being proposed to treat and prevent neonatal alloimmune thrombocytopenia. So not everybody does gene editing, and I just thought I'd take a couple slides to talk about that. Gene editing involves using a nuclease to make a double-stranded break in the genome, and that creates an opening into which templates can be introduced. A double-stranded break is an emergency for the cell. Your cell hates having its chromosomes broken, and so and we have DNA repair mechanisms that won the Nobel Prize a year and a half or so ago. One is called non-homologous end joining. It's non-homologous because you just shove some nucleotides in there right away, and you don't really care how many or which ones they are, just to repair the double-stranded break. And there's a ligase putting this in. When that happens, the gene usually goes out of frame if it's in an exon. If it's in an intron, it probably doesn't matter. And the other way to do it is something called, you make a homology-directed repair, where you take this break and you take advantage of it and you synthesize a template that has a slightly different sequence. And then that happens to join right in, and then you can actually modify the genome, and you can do this in all sorts of cells. So, gene editing has been going on for 20 years, but mostly inefficiently. And it's been guided to the site of cleavage by proteins that have specific DNA binding domains. So everybody's heard of zinc finger nucleases. These are proteins that bind to DNA and guide the nuclease, which is over here, to the right spot. but usually not. And then this was followed by something that's a little bit more effective called talins, and these are proteins that are derived from a bacterium that can also bind DNA and guide these nucleases to the right spot, but again, inefficiently. The thing that's cool about CRISPR is it uses an RNA molecule attached to a nuclease to guide the nuclease is sort of this fuzzy thing over here exactly to the right spot because it's based upon a 17 base pair sequence. As you know, 17mers mathematically only occur once in the genome if it was random. The genome isn't random, but if it were, a 17mer can only occur once. And so the off-target effects and the efficiency of actually driving this nuclease right to the right spot is very high. Meaning you only have to screen 1,000 clones to get 10 positive instead of 10 million clones to get 10 positives. And so you can sequence, so what you do is you take and you make this little loopy thing, it costs you about $80, and you put in the sequence of whatever gene you're interested in, and it attaches to this nuclease called Cas9. This will probably win the Nobel Prize in the next year or two. And then this sequence just guides the nuclease right to the site. And the nuclease has two cleavage domains, and you can actually mutate one to get a single-stranded break or a double-stranded break. A double-stranded break is shown here. And so cool things you can do with CRISPR, you can introduce stop codons into genes just by cutting it and letting it fill in. You can correct mistakes in genes. You can correct SNPs in genes. You can modify primary cells. You can modify the genes of animals. So you're no longer limited to transgenic mice because those are the type of cells that were easy to make. You can make transgenic kangaroos now. It doesn't matter. And you can modify and introduce plant genes to produce disease-resistant crops. If you're George Church at Harvard, you can make mammoths and Neanderthals. There's all sorts of things you can do with gene editing. And so what we decided to do is, and this has been around for a few years now, it probably became popular 2 or 3 years ago, and now many labs are doing this to modify their cell lines or their mice, is to modify iPS cells so they express rare allogenic epitopes and also fail to express class 1. So here's the beginning part of the GP3A gene. This PLA1, PLA2 polymorphism is in exon 3, and so we designed a couple guides that would cut the DNA here and here and remove this chunk. And then we designed a repair template that would hybridize over here and hybridize over here, but change the leucine to a proline. And so that was the whole purpose, is to remove this chunk and put in the chunk we wanted. And so and all you do is you take these guides and you make them, I think they're $25. You put them into these plasmids, which you can get from AdGene for $60. So this is really readily available to anybody with a credit card. You clone the guides. Yeah, Neiman Marcus catalog this Christmas will have these things. So you can you put these guides into a plasmid that contains Cas9 already, so it encodes the nuclease. And then you can either put in a selectable marker like puromycin, or you can put in GFP if you want to see which cells took up the plasmid. And essentially you take these plasmids, you put them into cells along with this HDR repair template. These are usually 100 to 200 bases, which you can now easily make on an oligosynthesizer. And then you put it into cells. And so the transfected cells in this particular case light up. You do a FACS sort and you take the lit up cells. And then you can either clone them out by limiting dilution or do a couple sorts. And at the end of the day, if this PLA2 happened, it introduces this nucleotide substitution creates inadvertently a restriction enzyme site. So if it becomes PLA2 and you add this enzyme, it'll cut. And so here's an A1A1 plus and minus the enzyme, no cut. And here's an A2A2 plus and minus the enzyme, it cuts. So this is an iPS cell line that we've now made PLA2, and you usually have to sequence it to make sure that nothing else, when nucleases are around, bad things can happen. So it might be perfect here, and then 10 nucleotides downstream have introduced some other error. So you sequence around this area, and at the end of the day, so you have these cells. So then what we want to do is grow these cells up. Now they're induced pluripotent stem cells, so they're very primitive. And for those of you who don't think about this very often, Shinya Yamanaka reported in 2006 or 7 in a Cell paper that you can take certain transcription factors, OCT4, SOX, KLF, and c-MYC, and put them into fibroblast cells from somebody's cheek. And you can introduce these things in, and you wait a few weeks, and these things go in and reprogram a differentiated genome all the way back to stem cells. Not just hematopoietic stem cells, embryonic stem cells. And so folks make cardiac muscle cells and nerve cells and gut cells, and you can, because once they're pluripotent, you can put them into various forms of happy juice with differentiation factors. And of course, we're interested in blood cells, and so are you. Jamie Thompson at the University of Wisconsin at Madison put in slightly different transcription factors. And so there are slightly different cocktails that can all go in and miraculously reprogram fully differentiated cells. You can get buffy coats, you can get fibroblasts, you can get all sorts of differentiated cells and turn them back into embryonic stem cells. And then once you have them there, like I said, you can go from there. So what we do is we use a protocol developed by Debbie French, who works with Morty Pons at CHOP. Is we take these iPS cells and we put them in certain types of media, and I'll show you what those are in a little bit, and we turn them into hematopoietic progenitor cells. And these things have red cell markers for Mohan and platelet markers for me, and then what we do is then you change the media and you put in certain things that drive them down towards the megakaryocyte lineage, and then at the end of the day you want to take, and we can get millions and millions and millions of these. You know, normally in the body a megakaryocyte spews out 1,000 to 2,000 platelets, and so this is happening every day, bazillions of them. And so we can do this very efficiently in plastic dishes. This happens, we can get plenty of megakaryocytes. We haven't been able to, we and others, mostly others, haven't been able to, I'll blame it on them, haven't been able to figure out how in culture to turn megakaryocytes efficiently into platelets. Most labs get about 10 platelets per megakaryocyte in culture. So this is a very active area of investigation in 3 or 4 labs, including Morty's. There was an RFA issued by the NIH a couple of years ago, and folks are working on this, and obviously you just have to recreate the environment that's in a bone marrow with all the growth factors and cytokines and proteins that it's sticking to, laminin and things like that, to get this to happen. So right now, and the goal is to make, you know, bags of platelets, which if we did it with current technology would be at $250,000 a bag, because right now we can only make enough platelets to transfuse and save the life of our fruit fly who's been in a car accident. So, but actually we can make enough to do flow cytometry and typing. So the way that we do this is we take these. It's a monolayer differentiation of iPS cells. You put them in these media for this number of days, and at the end of 10 days you can get hematopoietic progenitor cells, and then you switch the media over so that you can start making megakaryocytes. And you can follow this by flow cytometry. So early on, these iPS-derived ES cells have a couple stem cell markers, and they're double positive. When they get to this stage and they have hematoendothelial-like properties, they express CD31 or PCAM1, another one of my favorite molecules, and KDR. And then when they become HPCs, they express CD235, which is a red cell marker, and CD41, which is GP2B. And then when you put them into the megakaryocyte media, they express just platelet markers from that point forward. Not all of them, but these things, you can expand these greatly, and you can expand these depending on your protocol. And then they stop at a certain stage of differentiation and wait to make platelets. And so you can see here when we took these iPS cells, the iPS cells we started with were PLA1, which is 0.85 gene frequency, so that's, you know, you can type them. And when the ones we CRISPR edited which I showed you genotypically were PLA2, and by editing them it didn't screw up their ability to differentiate. And then you can see here's the original iPS cells. They bind to a PLA1 monoclonal called SC21. They bind to a human, these are the original ones, and they don't bind to a PLA2 antisera from a human. And then when you do the CRISPR, they still have GP3A, but they lose the PLA1 epitope and they gain the PLA2 epitope. So phenotypically, they've also transformed. And then we want to knock out HLA class 1. As many of you know, class 1 is a 2-chain molecule, a heavy chain and a light chain. This is the very polymorphic part right here. But to get to the traffic to the surface, it has to pair up in the ER with a invariant molecule called beta-2 microglobulin. So if you don't want this out there, you just get the gene for beta-2 microglobulin, you make a single guide RNA and you cut it and it goes out of frame, and then when this isn't expressed, this isn't expressed. And so you can see that this is done with DAMI cells, but the same thing happens with MEGs. The untreated DAMI cells have beta-2 microglobulin, they have class 1, and when you knock it out, knocking out is 2 weeks. You can knock out a gene in 2 weeks, easy. And journals will soon require you to prove that what you're thinking happens by doing this. Anyway, they become double negative. So essentially what we are doing is we're making a megakaryocyte or platelet bank and that have no class 1, which is our founder line, and then we just go through, just meaning, this actually takes a little bit of time because when you do homology-directed repair, it's a lower frequency event than just cutting and filling in. But what we hope to do is have all these major alleles and give them to Brian Curtis in our platelet antibody detecting lab, and then they can easily have these things that he can't get a hold of, all these rare antigens. So that's the first 2/3 of what I want to talk about today. And before I go on to the next part, are there any questions or comments about that part? Is it clear enough? What isn't clear? Everybody got it? Okay. So stay tuned. These things, we're just working our way through this table of these antigens now. So there's this project that it was initiated in Norway where they've typed 5,000 to 10,000 people, and they would like to treat neonatal alloimmune thrombocytopenia using the same type of thinking as we treat hemolytic disease of the newborn. And so, as you know, Rh-negative women who carry Rhd-positive fetuses are given an intramuscular injection of a human plasma-derived anti-Rh antibody to bind to this Rh antigen and make it immunologically invisible to this Rh-negative woman. So PLA, and it's called ROGAM or anti-D, and it's usually given at 28 weeks of pregnancy to prevent alloimmunization. And an Rh-negative mother who's not alloimmunized who hasn't been alloimmunized previously, usually receives a dose of ROGAM anyway after delivery. And several groups have proposed a similar strategy to prevent alloimmunization to PLA1, because it happens about 1 in 1,000 live births. But there's no animal model to see if this would work, so we decided to create one. And so what we wanted to do is create a humanized mouse model of this to examine the efficacy of some of these reagents that people are developing that will bind to the PLA1 antigen and mask it so that the mother's antibodies don't bind and clear fetal platelets. So as I showed you before, here the PLA1 is this leucine 33, but it's within this very flexible loop. And Emily Barron-Casella, a long time ago, did this study with a fragment of GP2B3A recombinant fragment, and she lined up the mouse sequence of this loop and the human sequence, and noticed that there were 4 differences between residues 22 and 39. And so here's the leucine, 33, and mice and rats have a glutamine there, and they also don't have a P here. And so the first thing she did is just change the SQ in the mouse protein to PL. And human PLA1 antibodies didn't bind at all. And she found out that she had to change this A and this D also. And when you did that, then, so this is 32, 33, 39, and 30, then PLA1 antibodies bound fine, just as well as they did to human 3A. So you have to humanize this entire loop, and then PLA1 antibodies bind. So the epitope is conformationally dependent. And so here's this APLD, you can see it here. And so what we did is we designed a homology-directed repair template of about a couple hundred bases with arms over here and over here, and then we changed the sequence in this little piece in the middle. And again, in exon 3, just put it in a guide to where it would cut here and open this thing up, and then this thing should go in and change the mouse loop to the human loop. And so, again, you just put the guide in, and you inject in the guide, this repair template, and then we make Cas9 mRNA runoff transcripts, and you inject it into zygotes. So you inject a couple hundred eggs, fertilized eggs, and then you screen the babies. And I had a grant due, and I was going to propose this as an aim. And I think the grant was due in January, and we started doing this in October, and the mother gave birth to 6 babies, and she ate them all. And so my grant was still due, and so we quickly re-injected a bunch of other eggs, and and this is a terrible mother mouse. She gave birth to 2 babies. One was dead, and the other one had converted. So, and so this one, I'll show you. We put in a sequence that would cut with BAM1. And so this baby mouse cut as it should have into a couple pieces, and when it sequenced it, it introduced these residues. And we call this the APLD mouse. And so that's a mouse protein that has a human PSI loop. All right, and in theory, this whole protein now should bind PLA1 antibodies. And so we took the mouse platelets from the APLD mouse and reacted them with a whole bunch of different NAI sera that Brian Curtis gave us from the platelet antibody lab, and some PTP sera. And whenever these sera bound to human 2B3A, they also bound to these mouse platelets, and they never bound to wild-type mouse 2B3A in black. So these platelets express this epitope, so that's good on the surface. And then what we did is we took a wild-type mouse and we immunized her with APLD, so she would make an anti-PLA1 antibody, a little bit of adjuvant and some platelets. And sure enough, over a period of weeks, and we took about 6 or so, I can't remember how many colors there are, but we took 6 or 8 mother mice and we injected them with APLD platelets. And over a period of time, this is flow cytometry, they made a whopping antibody. 35,000 by flow cytometry, so negative is way down here. And so that was good, and then we wanted to see if that antibody was binding to APLD GP3A. And so we did some Western blots. So mouse platelets don't react, a couple different mice, and APLD mouse platelets, you dissolve them, add them to this antibody, and it's a specific antibody for APLD GP3A. So that's good. And then we took this mouse who'd made antibodies, and we put her in a cage with this guy, and she made babies. And so here's a wild type pairing, and there's the platelet count of, these are all the babies. These mice have, it's an F1 C57 black BALB-C hybrid mouse. BALB-Cs make better antibodies than C57s do. And they have litters like of 13 mice, which is like double what other ones do. But anyway, when the father was APLD and the mother was this immunized wild type, the babies were born with no platelets. And you don't need error bars. I like type of data where you don't need error bars. And so I think one of the mice here who had a whopping, I think we took her out and took her spleen and are making monoclonals, but we took the ones with highest titers, and they gave us these, and those with a lower titer still gave birth to mice that are thrombocytopenic, some of them severe, some not so severe. But that mimics what happens in the human disease too. Sometimes the babies are born with 50,000 platelets, and sometimes they're born with 5,000 platelets. And so we have this, and now what we're, so this is like February's data, so way not published. But what we hope to be able to do is infuse into her a reagent that can prevent her antibodies from binding to these fetal platelets and bring the platelet count back up to here. And then I think we'll really have a therapy for this disorder. And there are a number of little tricks you can play to introduce antibodies into the mother mouse that will prevent fetal platelet clearance that we've shown in other in vivo models. So sort of the summary of all this is that you can combine IPS and megakaryocyte and platelet technology with gene editing technology to generate knockout and knock-in platelets, and we call them designer platelets, and we're trying to make a bank that'll be actually useful. Again, all founded on years and years and years of basic science studies. The ability to generate a large number of these for purposes of transfusion is still far off, and it's going to require an improved basic science understanding of the factors responsible for differentiation and platelet poiesis. And I think when you do creative editing of the megakaryocyte genome, you can also do things that will enhance thrombopoiesis, enhance the function of platelets. So there are all sorts of things you can do with known platelet genes to make the platelets more and better. And we have a person or two working on that as well. And then, you can now make these mice that are customized. You can put antigens on them. You can humanize regions that you need. And we hope that this will be a valuable tool to assess some treatment options that are being developed in Norway and in other places for women at risk for developing an alloimmune response against their unborn babies. And with that, the CRISPR team, Nanyan is the designer platelet lady. She's in 7 days a week. iPS cells are tricky to grow. They require constant attention and babysitting. And Hui Ying made the APLD mouse. And this is sort of the rest of the lab. And so with that, I'm happy to stop and take any questions. So thank you. Yes, sir.
It's very important to do. Can you get, how many platelets do you need for typing in a regular typing lab, but Curtis, and how much can you really generate with these iPS cells?
You can generate, so if you want to do flow cytometry, you only need, tens of thousands of cells, and we can make millions right now. In fact, these HPCs, which expand for a long time, express 2B3A, and so for the antigens that are on that protein, they're already positive. So we can already do flow cytometric detection of all these different antibodies quite easily. If you need a platelet, that's a little bit tougher, but right now we can make a lot of megakaryocytes, and that seems to be positive. So what I hope to be able to have are little vials of frozen cells that you can expand when you have these rare, tricky cases and do it. But so anyway, flow cytometry, well, I showed you flow cytometry that they're positive for PLA2, and that's fairly easy. Like I said, platelets for transfusion, we're not going to replace donor-derived platelets with this technology. But if you need specific antigen-positive platelets for specific women under particular situations, I think there's hope for that, but probably not for another 5 or so years. Folks have to get better at making platelets in a bag. Yeah.
So I had a question. So what is your goal in the iPSC cell-generated platelets? Is your goal to actually generate units, or is there scope for reconstitution of the clinically affected patient themselves?
Yeah,
let's
so
say the progenitors.
you can't make units yet. You can make little tiny bags.
Right.
Right. But so the goal right now is to make enough for diagnostics, and we can already do that. And you can also do investigative things. So there are glycoproteins where the carbohydrates can differ. You can play with that. We can make cells where we knock out negative regulators of platelet function and make platelets function better, and we can look at that. So we can do investigative things. We can do diagnostic things. Therapeutic things, like I said, are a ways off, and it requires, we don't know enough yet, to understand all the different things that happen to a megakaryocyte that allow it to make 1,000 platelets per cell. And so, the science still has to go forward in parallel with these translational
One last point
things.
that comes up with any iPSC-generated product. How do you plan to actually prove that your platelets are platelets?
Yeah, so they'd have to be made in a GMP facility. One of the things when you make iPS cells and you're going to make a nucleated cell, there's always the chance that some undifferentiated cell stayed around. And so you could transfuse that, and let's say you get to the transfusion stage 5 or 7 years out, and there could be some iPS cell that could then get into a place in your body and differentiate into something you don't want, where you don't want it. So with platelets, there's no problem because you can just irradiate them. We don't have a nucleus, so go ahead. So platelet transfusions will be easy. Other types of cells that come from iPS cells have the danger of having an undifferentiated cell sneaking in with your differentiated cells. So that's a good question. Yeah. Yeah, Ed.
Really interesting. I'm wondering, however, if you could just remind us what the normal function of the PSI domain is, because there seems to be a fairly high frequency of variation in that region, and I'm wondering why it's persisted.
The other nucleotides that I showed you that are polymorphic in GP3A are not in the PSI domain. And so, and the function of the PSI domain depends who you ask. There's this guy in Toronto who thinks that it has protein disulfide isomerase type activity. and Bruce Fury and Barbara Fury also think that PDI activity is important in rearranging disulfides in proteins like tissue factor and turning them from cryptic to active. But the PSI domain isn't terribly functional normally, and the polymorphic forms of PLA1 and PLA2 seem to function similarly, so that SNP doesn't change the overall function of the molecule, although there were probably 100 papers saying that PLA2 is at risk for cardiac disease, but after 10 years of research, it didn't hold up. But anyway, that particular SNP doesn't seem to be functionally important. And you know, all your genes are polymorphic, like you getting out in the sun over a period of a couple million years, you know, and everything, but it doesn't mean it's immunogenic. So most of your polymorphisms aren't recognized by the immune system. So there's a difference between polymorphic and immunogenic. Immunogenic means it has to be chopped up, it has to fit into the groove of some HLA class 1 or class 2 molecule and present to a T cell receptor, and that doesn't happen most of the times with polymorphisms. Yeah.
Thanks.
Yep.
Small token of our appreciation.
It's a bag.
Gee, thanks. Now he's going to come rescue the computer again? Okay. So it's my great pleasure to introduce our next speaker, who is Dr. Heather Pidcoke. Heather is coming to us from a very interesting background. She's an MD PhD, having done her medical training at USC, and then her PhD degree at UT San Antonio, where she went from there to become to work for the U.S. Army Institute of Surgical Research, where she was the deputy chief. And so she's bringing to us this interesting sort of surgical perspective, also with the military theater in mind. Heather left the role in the military to go into the private sector, into a job where she has interaction with some of the people in this room, because she is the director of global clinical safety and pathogen reduction technology scientific affairs at Terumo BCT. And so it's my great pleasure to welcome Heather to give a presentation after Ed puts her slides in.
Thank you very
It's my
much.
role here.
Thanks, everyone. So I'll be talking about refrigerated platelets and new opportunities for improving the care of bleeding patients. First, I have a boatload of disclosures. First, the Army does not want to claim anything I say here today. And Terumo BCT also does not want to claim anything I have to say here today. But despite this, they are collaborating to bring these products to the market, so my conflicts of interest are legion. So where did this project start? Well, during the wars in Iraq and Afghanistan, the number one cause of preventable death was hemorrhage. And so the challenging thing in those austere environments was the supply of platelets, because products came from the U.S. There, but as you know, platelets are only stored for 5 days, and so this created a situation where it was very difficult. And it was also considered risky because of the potential prevalence of infectious agents. So platelets stored at room temperature were mostly unavailable. There's also a very well characterized platelet storage lesion. So the current standard of care is general agitation for 5 days. There are longer in vivo circulation times for room temperature platelets, which is why they were adopted, because at the time in the 70s, the idea was that 80% of platelets were being transfused to cancer patients who were not able to produce their own platelets, but there were some fairly significant disadvantages, including increased risk of microbial contamination, as I mentioned, the storage lesion, the short shelf life, And there was also a robust discussion at the time about whether these platelets would be suboptimal for trauma patients because they did not react as well to agonists compared to cold stored platelets. So despite that discussion, room temperature stored platelets were adopted. So the main reason why there's longer in vivo circulation times for room temperature platelets is that cold stored platelets undergo a shape change and express unusual receptors on the surface for a resting platelet and are recognized to be altered by the spleen and the liver, and they're taken out. So the question, though, is how are platelets used today? Well, if you look at the ABB report of how platelets are used in the United States, you can see that the biggest category by far are the oncology patients, but they still don't come anywhere near the 80% that people thought before. And if you look at the surgical services, they are all split out and not treated as an aggregate as the oncology services are. And in fact, the same is done with the medicine. So if you realigned everything and looked at indications rather than services, you would probably find that there's probably more like a 50-50 split between patients who receive platelets because they're bleeding versus patients who receive platelets because they're thrombocytopenic. So what about the hemostatic function? Well, in this study, healthy patients were given aspirin to create a platelet lesion, and then they were given either cold stored platelets or room temperature stored platelets, and the activity for the room temperature platelets was about half of that of the cold stored platelets. So there's no recovery of the aggregation response to ADP stimulation post-transfusion after even 2 units of platelets if you give room temperature stored platelets in these healthy patients who took aspirin and Plavix. So at the Institute of Surgical Research, we looked at platelet function in cold stored whole blood to try to get an idea of what the platelet function was. We looked at days 1 through 7, 10, 14, and 21, and assayed platelet aggregation, thromboelastography, microparticles, and platelet activation. So what we found was that there was a marked difference between the response to agonists and the ability to aggregate in the cold stored platelets compared to the room temperature. The cold are shown on the top in purple. That was in whole blood, and so then we repeated those studies in platelet components, and we found a very similar picture. The bottom graphs here show platelets that were reconstituted with blood and plasma to try to simulate what happens in vivo, because some people theorize that room temperature stored platelets are somehow reactivated and made to work better once they're transfused. We didn't see that effect in this in vitro simulation, but again, this was in vitro work. So why do they work better? Well, one of the reasons is that their clot strength is much better in the cold stored platelets. So on the left here, you can see that there's fresh platelets shown in green, and if you compare them to room temperature stored platelets in red versus cold stored in blue, you see that the clot strength is much better preserved in the cold stored platelets. We wondered if plasma was contributing to this loss of clot strength, so we took a look at fresh plasma versus cold stored or room temperature stored, and we saw that there was almost nothing preserved of the plasma contribution to the clot strength. So we looked at breaking strength, which also confirmed the thromboelastography results, showing that the room temperature stored platelets had about half of the breaking strength of the fresh platelets, whereas the breaking strength was preserved all the way out to 14 days in the cold stored platelets. And then because we wanted to know why this occurred, we looked at cross-linking density, a measure of cross-linking density, which is the slope of the breaking strength curve, and we found that, again, the cross-linking was much higher in the cold-stored platelets. And this can be seen by SEM, where platelet strength is a measure of the thinness of the fibers and the density of the fibrin mesh. You can see that in the fresh platelets, there's sort of a medium-sized fiber with an intermediate density of the mesh. That is the thickest fibers and the loosest mesh is seen in the room temperature stored platelets, whereas the cold stored platelets actually have higher density and thinner fibers. And this is related to the function of factor 13 in cross-linking these fibers. So then we also wondered about the mitochondrial activity within these cold stored platelets and whether the mitochondria were similarly preserved. And so you can see that the magenta shows intracellular mitochondrial reactive oxygen species, which is a marker of mitochondrial damage, and it's much higher in the room temperature stored platelets compared to the cold. This is a measure, this is a measure of mitochondrial capacity. So the closer you are to 1, the lower your mitochondrial capacity is, and the less potential you have in the mitochondria. And you can see that the cold stored platelets, again, have better mitochondrial capacity compared to room temperature. So how does this correlate with room temperature functional impairment? Well, the correlation is pretty strong between the mitochondrial function and the ability to aggregate the clot strength as determined by ROTEM, and even in the clots breakdown. So one question that we wondered is the cold-induced priming of platelets that causes them to change shape and makes them cleared by the system. Could this be related to apoptosis? As a result, we took a look at several different forms of caspase, and in every case, the caspase was higher in the room temperature stored platelets compared to the cold, so this was not a function of apoptosis. And then the other question that naturally comes to mind when a platelet is primed for activation is whether that's going to participate in clot in a pathological form. So would these platelets respond to physiologic inhibition? So we took a look at these platelets and exposed them to physiologic inhibitors. So you can see that the bars on the left at the bottom are the uninhibited platelets. And then we have 3 groups. One is the room temperature and the cold in the middle and the cold on the right. And you can see that the room temperature really, there's not much of a response of the platelets to agonists to begin with, but that there is quite a robust response in the cold platelets. However, the cold platelets are able to respond to the inhibitors quite well. So this was confirmed with non-physiological inhibitors such as cytokinase B and eptifibatide. The other question would then be, what about intracellular signaling? Is the response that you see to the inhibition a result of specific signaling, or is there something else going on? So we looked at cyclic AMP and cyclic GMP response levels to inhibition and found that, in fact, it is intracellular signaling that is producing this effect. So the other question is, what about bacterial growth in these platelets? Well, we took a look at that as well and found that the bacterial growth in spiked platelets was inhibited, whereas there was robust growth within the room temperature samples. So now there are some issues with storing platelets at refrigeration temperatures in plasma. One is that the platelet count decreases over time. It forms visible aggregates, and that's been noted since the 70s, and that there's lower recovery and survival times. So this is demonstrating those effects. You can see that there are white fibrinous clumps in the cold stored platelets and plasma, and that this is reflected in the platelet counts that are quite lower after day 3 and day 5. So why do these aggregates occur, and how do we prevent them? So the GP2B3A receptor, in that partial priming of these cold stored platelets, more of it gets moved to the surface and it becomes activated, and as a result, if you have fibrinogen in the medium that the platelets are stored in, it's going to cause clumping. So we hypothesized that if we eliminated or reduced the fibrinogen in that storage solution, that we could prevent that platelet aggregation formation when stored in refrigeration temperatures. So we first took a look at platelets in platelet additive solution, and this was a 100% platelet additive solution. And we found that, in fact, you can prevent that aggregation just by changing the storage solution. And you can see that in the platelet counts that are stable at days 3 and 5. The problem with that is that is not approved for human use, because room temperature platelets in 100% pass solution don't do well because they're metabolically active, and so they use up all the sugar, and then the platelets perform very poorly. So we changed that to 65%, 35% plasma, and found that, in fact, you're still inhibiting that aggregation, and your platelet counts remain stable over time. So here's a comparison of 4-day platelets and plasma at day 5 on the left, and 15-day cold platelets in 65% pass, 35% plasma solution, and you can see that even at day 14 there's no visible aggregates. So what about platelet activation markers? Well, there's no difference between room temperature and cold stored for P-selectin. Interestingly enough, the phosphatidylserine exposure, which is a marker of cell death, is higher in the room temperature stored platelets compared to the cold stored. So microparticle formation is very similar between the two, certainly not worse for cold platelets. And then how do these platelets function in aggregation studies? So the 22-day platelets, you see an uptick in the collagen, and that's a factor of the shape change. When platelets change shape, you see an decrease in aggregation, which shows as an uptake. But in the cold platelets, that's gone. However, they respond to aggregation at day 5. So here is some data showing aggregation response over time. You can see that there's very rapid loss of the aggregation response in the room temperature platelets, reflecting the storage lesion, whereas in the cold platelets it's preserved almost out to day 22. One of the things that we have heard from our colleagues is that in physiological circumstances, you don't have one agonist at a time, that you have a combination of agonists, and that if we were to do these aggregation studies using combined agonists, that we would see preservation of the room temperature platelets similar to the cold platelets. However, we did that here. This is a combination of collagen and epinephrine, and you can see that there's not much preservation in the room temperature stored platelets, but very good preservation in the cold. So to summarize this part of the talk, we saw aggregation responses up to 15 days and some even out to 22. We saw stronger clots when platelets are stored in the cold, and they maintain their mitochondrial function, and they can be inhibited by physiologic antagonists. So what about the platelets in PASS? We found that they maintain their platelet counts and don't form visible aggregates. They're functionally primed in static and flow assays, and they have microparticle levels similar to room temperature. They display aggregation responses at least out to 15 days, and in some cases out to 22. So we hypothesized with this program that we can produce a product that is better in that it has better hemostatic properties, which are important for bleeding patients, that they're safer because they lower the risk of bacterial contamination, reducing testing costs, and they're also going to be able to be stored longer because they remain physiologically active out to 15 days. So how do they perform in vivo is the next question. Well, in this rat model, these rats are given a micro-injury intravascularly, and then intravital microscopy is used to look at the injury, and then they're transfused with platelets from GFP-labeled rats. And you can see that the green transfused platelets are incorporating very well into the wound, and this is 5-day stored platelets in the cold. So then our next research question is, how are these going to perform in humans? There's a group of colleagues in Norway, in Bergen, who are looking into this question in a complex cardiac surgery study in which they are comparing cold stored single donor platelets to conventional platelets. So in this study, they are looking at 5 days right now. So all the data that I'll show you will be for 7-day stored platelets in a platelet additive solution called TPAS Plus, which is the another name for it is SSP Plus, just from a different manufacturer. In their study, the primary endpoint is in vitro coagulation responses by looking at aggregation and multiplate, but they're looking at secondary endpoints, including immediate blood loss. So they're tracking that by measuring the chest tube output for the first 24 hours after surgery. And they're looking at biochemical and hemostatic changes during the peri and post-operative periods. And they're also looking at short-term and immediate complications. So these patients, at the time of reversal, when bypass is being taken off, if platelets are needed, they will be given either the cold stored platelets or the room temperature control, and then followed for the first 24 hours, and then for a longer period for the complications. So this is a result of the interim analysis, so it's not the final data. This was presented by our Norwegian colleagues at AABB. You can see that the aggregation response in the platelets is better than the room temperature for all of the agonists shown here, and that effect is significant for ristocetin. The platelet count is a little bit lower in the cold store platelets, as would be expected, because this is in vivo platelet counts for the patient, and as we would expect, that level would go down faster in the cold stored platelets. When you correct, platelet count obviously affects aggregation response. You can't break up the two. So when you divide the aggregation response by the platelet count and in effect correct for the effect of count itself, you can see that the cold stored platelets either perform equally, or in the case of ristocetin, which is a measure of flow-dependent aggregation, it's much higher. So the interesting finding for this study is that there is a difference in the chest tube output compared between the cold platelets and the room temperature. At the time that this analysis was done, this effect was not significant, but what I've been told is that it looks like it is becoming significant as they get adequate numbers for power in the study. Obviously, this study is way too small. It's 20 per arm to make any definitive conclusions about it, but the one thing we can say is that certainly chest tube output with the cold store platelets is not worse than the room temperature. So the study conclusions to date are that we can possibly improve platelet aggregation response and other measures but if not improved, they're at least as good. So you could take a longer stored platelet, hopefully, and produce the same effect, although that study has not gone into the next phase, which is to look at the 14-day platelets compared to the control. But the purpose of the study is to evaluate these platelets for non-inferiority. Again, not something you can prove with a study because it is a pilot study just to look at it in preparation for larger randomized controlled trials. There's no thromboembolic events that were observed within 24 hours after transfusion of the cold platelets. The transfusion of cold stored platelets are efficient for the treatment of post-operative bleeding in complex cardiac and thoracic aortic surgery, so they were at least able to control the bleeding as well as control, if not better. So these platelets have been started to be used clinically by the U.S. Army. They're being collected at Bagram Airfield in Afghanistan, stored for 5 days. And the validation process has been completed. All of the units that they use there at Bagram, because cold platelets in platelet additive solution stored for 14 days are not an FDA-approved product, the Army cannot use them. So what they're using is platelets and plasma up to 5 days with all the attendant issues of platelets and plasma. But still, all of the units that they collected passed acceptance criteria on day 5. They don't pass swirl because, again, they have had the shape change, and so the swirling characteristics are different. But otherwise, they did fine. One thing that they did find out was the theory at the time was that you should be able to store them in any position because in the cold, they're not subject to the same problems of room temperature stored platelets, of metabolic activity resulting in the need for oxygen, resulting in the need for agitation and for the bag label being down. Well, something else is going on because both Mayo Clinic, which has also started using these platelets routinely for day 3 storage, and this Bagram Airfield project found that if the label is stored up, for some reason the platelets don't do well and you see much more aggregates forming, which is something that we need to investigate and figure out why. However, they were successfully validated. So here's the validation data from the Afghanistan project. So in routine use, 4C platelet products have been transfused since January of 2016. And this was done in Afghanistan. 7 patients received cold stored platelets, one of whom received 4 units over a 3-week period. They received other types of blood products as well, of course, as part of their resuscitation, and no negative outcomes were noted. The logistic burden was decreased because they were able to dispense with culturing for bacterial contamination, and they did not use platelet agitation, which also helped them quite a bit. So as I mentioned at the start of the talk, the DoD is working with Terumo BCT to try to get approval for platelets in the cold. So this is the team at the Institute of Surgical Research where I worked that worked on these projects, and this data is not mine but belongs to the group. Any questions?
Thanks. Well, people are getting their thoughts organized. I'm curious about, so the FDA licensed 3-day cold storage like June of 2015 or something, and those are in plasma?
Yeah, so that was actually an exemption. The cold stored platelets were the routine care back in the 60s and early 70s And they were, platelets in general were only approved for 3 days of storage at the time. So that has never come off the books. In fact, when I mentioned that discussion that happened in the 70s, there was a conscious decision not to take them off the books. However, platelets have undergone a lot of changes since then, and during that time, the ABB never bothered to create any standards for cold stored platelets because no one was using them. So now that there's interest again, Mayo Clinic applied for an exemption to use apheresis platelets in plasma because in the CFR of the FDA, it says very specifically that they are whole blood derived. So FDA looked at that and at first refused. So Mayo persisted, and with a little more data and more academic interest in cold platelets, FDA reconsidered the following year and approved it for the treatment of active bleeding. And so that's where that came from.
Thank you. Obviously, if it was more than 3 days, it's a very intriguing inventory management solution for blood operators.
Okay, fascinating and very encouraging. So we're going to finish up with 2 types of platelets in the blood bank, presumably.
Yeah.
We're going to have prophylaxis, where we're looking for some form of longevity, and hemostatic platelets.
Yep. Yeah, and the inventory management, shipping characteristics, those kinds of things can't be discounted as an advantage in terms of supplying platelets to hospitals that are in regional areas of shortage. So there's no platelet shortage in the U.S. At least. I'm not sure about Canada. If you look at platelets across the board, but if you look at regions, there are some regions where they're incredibly in short supply. An example would be MD Anderson, which vies for the top spot in the U.S. for cancer treatment. They have such a critical shortage of platelets on a daily basis that they are scrambling at the beginning of the day to figure out how they're going to supply their need at the end of the day. They have told us that they do not ever transfuse prophylactically at MD Anderson because they absolutely cannot. They don't even routinely give an entire 6-pack of platelets. Most of the platelets are whole blood derived, although they get a significant amount from apheresis, but they don't even give a full transfusion dose of platelets because they can't, and so they give 2-packs or 3-packs instead of 6-packs. So places like MD Anderson will be much better able to supply their patients if other places that have relative platelet abundance have the opportunity to ship them. And then there's, of course, platelets continue to be higher in bacterial contamination by a power of 10 compared to refrigerated blood products.
Other questions? No? If not, thank you very much, Heather.
Thank you.
I'll talk, you push buttons. Thanks very much, Heather. That was really very interesting. Our next speaker is Shabani Patti. Shabani is the Scientific Director for Cellular Therapies at Blood Systems Research Institute, and she's also an Assistant Professor at the University of California at San Francisco in the Department of Laboratory Medicine. Shabani, in addition to all of her very interesting work in cellular therapies field, has done some really interesting work looking at I think sometimes a long forgotten area of transfusion medicine, and that is what we do with transfusion plasma. So I've asked her to come share some of that work with us today, and she's giving us a talk, which is the title you can see on the slide right there. So Shabani.
Thank you, Dana. It's really wonderful to be here today. I thought today I would sort of present to you a more conceptual talk of some of the work we've been doing over the past couple of years, trying to understand how component-based therapy works in trauma and other indications aside from hemostasis. And so with that, I'll start with it's interesting, I don't know why there's a black box there. But anyway, just to give you a little bit of background, there have been a number of clinical studies, both in the civilian and military sectors, suggesting that administration of higher FFP to platelet to RBC ratios in massively transfused patients is associated with a survival benefit. And in many ways, these findings have really formed the principles of what's called damage control resuscitation in trauma. A number of papers which were generated early on, starting with a seminal paper in 2007 by Borgman, Spinella, and Holcomb and colleagues, showed that the ratio of blood products that was administered to massively transfused patients was critically important in terms of outcome and mortality. And as you can see here, again, this was a retrospective study showing that as the ratios were closer to becoming more balanced towards a 1-to-1-to-1 plasma to platelets to RBCs, that you could see a very significant drop in mortality. Since then, there have been 2 trials, PROMPT and PROPER. PROPER was actually completed last year, which was a prospective randomized trial to study the optimal ratio of platelets to plasma to RBCs. And in this study, it was basically a large pragmatic multi-center RCT with 12 sites, 680 patients. And the ratios that they tested were quite narrow. It was just 1-to-1-to-1 versus 1-to-1-to-2. And it's a very long story of why they were not able to spread the ratios further. And in part, it was due to the fact that practice had changed in many of these centers already, with balanced ratios being utilized or essentially in practice. So in this particular trial, the primary outcome was mortality, and there was no difference in overall deaths at 24 hours and 30 days, but it was interesting that they found that deaths from exsanguination were less at 24 hours in the 1-to-1-to-1 group. And during the protocol period, when 1-to-1-to-1 versus 1-to-1-to-2 was actively being administered, there were actually less deaths in the 1-to-1-to-1 group. So interestingly also, there was no increase in complications in the 1-to-1-to-1 group in the first 24 hours. So with that in mind, we have realized over the past couple of years that many of these findings call for mechanistic studies into why balanced ratios with increased use of plasma and platelets would provide a survival benefit. So if you actually take this is a picture that was sent to me by a colleague of mine, Martin Schreiber, who works at OHSU. This is the floor of the OR. Essentially, this is the amount of blood products that are actually getting transfused into a single patient. And one recognizes that all of these have biological effects. Yes, they're repleting volume lost of blood products. They're also providing hemostasis. But how are they actually changing the patient that's receiving them? So with that in mind, we, over the past few years, have asked the question, how do plasma and platelets work in damage control resuscitation in trauma patients? So we formed a very basic hypothesis, and this was that if we prevent or repair endothelial dysfunction early after traumatic injury, that we could improve outcomes. We furthermore thought that plasma and platelets in DCR and transfusion and trauma actually repair endothelial injury and dysfunction, and that this could actually lead to improvement in outcomes, and especially outcomes that are related to organ injury, such as multi-organ failure. So with that in mind, we embarked on a number of studies, and in many of our studies that we carry out in the lab, we've really targeted the vascular endothelium. In many ways, you can see it as an organ in itself. It is heterogeneous throughout the body, but communicates systemically. And it's really the platform on which a number of these processes we're all discussing take place in both health and disease, everything from coagulation to inflammation, tissue oxygenation. So with that in mind, we have really focused in upon an area, and there was a new term that was coined, which has been utilized over the past year or so, called the endotheliopathy of trauma. It's loosely defined as 3 main components contributing to it, and that is dysfunction of the immune system, coagulation disturbances, and disturbances in endothelial function. And of course, no one knows which comes first and which is the initiator of all the pathological disease. But in a narrow fashion, we've really focused in upon vascular stability. So this is our working biological model, and if you think about it, a blood vessel, when it's in homeostasis, it's not injured, all of the junctions that hold the cells together are actually intact. There's no edema in the interstitial space. The lymphatics are working well and taking up all the extra fluid. If you look in hemorrhagic shock, there's a number of insults going on aside from the site of direct injury. We have a systemic insult to the vasculature, which is caused by hypoxia. A number of cytokines and chemokines and growth factors are released. You have increased vascular permeability, increased cell contractility, extravasation of a number of proteins, and also leukocytes into the extravascular space, and also decreased uptake of fluid by the lymphatic vessels. This actually is exacerbated by fluid resuscitation with crystalloid fluids. So you end up with increased edema when you resuscitate with fluids like lactated ringers or normal saline, and it actually initially might bring up mean arterial pressures and replete volume, but the long-term consequences of crystalloid resuscitation are actually organ injury, increased inflammation, and detrimental effects, which we believe are repaired by plasma and platelet transfusion. So that is really the concept behind all of the sort of endeavors that we've embarked upon to study the effects of plasma and platelets in trauma. So this was the first start of some of our studies that we had, where we were investigating the effects of plasma on vascular permeability. In this particular assay, it's a very simplistic assay. We were looking in vitro at how plasma might affect the the endothelial monolayer in terms of the amount of leak that one sees. And this is all leak that's induced by VEGF. We've also done it with hypoxia. And what we saw here very clearly, which is day zero plasma, which was freshly thawed, in a dose-dependent fashion actually decreased endothelial permeability. We saw also there seemed to be, for this particular donor, a storage lesion that at day 5 that we did not see as protective effect on vascular leak. And although day 10 plasma is not used clinically, we actually saw that day 10 plasma actually instigated vascular leak. So with this in mind, and I actually would like to mention this is not the case for all plasma donors, so there is donor variability in how they age. If we look at about 10 plasma donors, only 2 will actually age in this fashion. Most of them retain their activity over the period of 5 days at 4 degrees. So we have a number of models in which we're able to look in trauma at vascular leak. One of the models that we use is a hemorrhagic shock and trauma model in C57 black 6 mice. And in this model, we subject the mouse to a controlled hemorrhage, which is for a period of 90 minutes. We hold them at a mean arterial pressure of 35 millimeters of mercury. And after that, for 10 minutes, we resuscitate the animals with our fluid of choice. These animals are actually also subjected to a second traumatic injury, which in many ways is the second hit. And this traumatic injury is generated by a laparotomy, a central-line laparotomy, and then sort of gentle agitation of the bowels. And so with that in mind, we're able to look at a number of different outcomes in these mice. And the first thing, the lungs in many ways are the canaries in the body. It is a canary that sings when there is injury. And the lungs are the first in many ways to get injured by any systemic insult. And what we found clearly is that these are sham animals. You can see histopathologically shock generates characteristics of lung injury. Lactated ringers, which is a crystalloid fluid, seems to exacerbate this injury, and plasma-based resuscitation attenuates it. So this, in essence, is also reproduced by quantitative measurements of vascular leak. So we're able to look at leak in the lungs in these animals. We administered to them a dye called Evans blue dye, and that binds to albumin. So the amount of vascular leak in the lungs we see here by shock, instigated by shock, is actually attenuated by FFP transfusion and not significantly by lactated ringers. We also see that there is a decrease in inflammation in the lungs. findings. This is MPO, which is representative of neutrophils. We see a drop in FFP transfused animals and no significant drop in crystalloid transfused animals. So with this in mind, we actually have a number of ways which we're able to look at leak aside from Evans blue. We are able to look at leak of different sizes of proteins. This is using infrared tagged dyes. This is essentially lungs from a shock animal. You can see that this is a dye of 10,000 kilodaltons, so smaller than albumin. And as you go up onto the red scale, it means that you have increased leak. You can see very clearly visually that in the lungs of animals that are resuscitated with fresh frozen plasma, that this leak is attenuated. And here it is depicted quantitatively. You can see here clearly there's a drop with FFP, and lactated ringers, the difference is not significant. So with that in mind, this was sort of a quick fly-through of our plasma-based studies. We have embarked upon a number of studies now to really understand what is it in plasma that is actually having these effects. What are the proteins that are active? And it turns out we have an extensive effort to fractionate plasma now, and what we found, I can tell you a few of our preliminary findings, is that it is not linked to the coagulation proteins. So it is definitely linked to proteins that are in the small range, and also a number of small peptides. So, and there is another study we have ongoing right now, which is to try to understand which of these peptides actually are beneficial and which are detrimental. So like any mixed bag, nature provides a balance, and some of these proteins actually seem to be protective to the vasculature, and some of them are actually detrimental. So really the idea, in terms of vascular leak, the idea is that it's a balance between these proteins, and perhaps that balance gets thrown off in injury. So with that in mind, you can ask the question, and we get asked this question all the time, so who cares about vascular permeability? I've been asked this on grants and also on papers, and so I'm like always sitting here trying to defend it. Hey, the vasculature, it's really important. And so, it's simplistic. Vascular permeability results in organ edema. Organ edema results in organ injury and inflammation. FFP definitively attenuates the start of this process and repairs organ damage. I'm not showing the data from other colleagues, but Hassan Alam, who is a trauma surgeon up at Michigan, has shown very clearly that if you transfuse animals, he's done large animal models of swine traumatic brain injury with fresh frozen plasma, cerebral edema is attenuated, and so is inflammation, and neurocognitive function is improved. So the bottom line here is that leak is important. There is no therapeutic currently in use to actually attenuate leak, and we believe that you could probably address a number of pathological conditions just by addressing vascular permeability. And if you think about it, actually in the lungs, the distance between, this is just a very simplistic way to look at it, the distance between the alveolus and the capillary for gas exchange is less than 1 micron. Any small amount of edema within that space there is going to cause effects upon gas exchange. So with that in mind, we feel like we are justified in investigating this area. So hopefully I've convinced some of you. So then the next step that came in, some of the questions that we've been addressing in the lab really pertains to platelets. And Heather did such a nice job of talking about cold stored platelets and hemostatic effects. We've really looked back, and this is not new. This was discovered a long time ago, that platelets have effects outside of their effects upon clot formation and hemostasis. And in many ways, they are the most critical regulators that are circulating that hold the endothelium and the vasculature together. If you think about it, so in the 1800s, actually Bizzozero, who's an Italian physician, discovered the role of platelets in the hemostatic role. But then in 1940, Danieli, who was another Italian physician, really showed that platelets had a very large role in preserving barrier function of the endothelium. And then further, the person who really developed this was the late Judah Folkman, and he showed that platelets maintained vascular stability were absolutely necessary to maintain function and prevent organ edema and bleeding in multiple models of injury. And so with that in mind, we asked the question, and this typically is not a question asked about platelets in terms of their function. So we worked on this for a couple years, and these two papers were published. I'm going to tell you a little bit more about the first paper, which really talks about platelets, but also we embarked upon some endeavors into understanding the difference between 22 and 4C platelets on these particular outcome measures. I'm going to mention just a little bit about what holds the endothelium together. So if you look at these are two endothelial cells. If you take a look, there's a number of proteins which actually hold the endothelium together: claudins, junction adhesion molecules, nectins. Now, one of the sort of major sort of binders of the endothelium is actually the adherens junctions. Adherens junctions are formed by V-cadherin binding from one cell to the other. And we can actually, in a very nice way, visualize these junctions. This is actually endothelial monolayer in vitro. These are pulmonary endothelial cells, and what we've shown is that if we stain for V-cadherin, you can see here that it forms a very nice border around the cell. Now, this is actually a non-leaking monolayer, and if you expose a monolayer like this to hypoxia, you actually sort of knock out all of these junctions. If you add platelets to this monolayer, within 15 minutes you actually reconstitute these junctions. So that is how critical the role of platelets is in maintaining vascular integrity. You can see this is a 15-minute and then it's fixed, so it's a very rapid effect. And these are platelets with no plasma, so we've removed the plasma from here because plasma has the same effect. So with that in mind, I'm actually going to jump 3 or 4 slides forward because I realized I put the model, how this model is run, the slide for that a little bit ahead. So I'm just going to enable me to describe it to you. We actually are able to look at endothelial permeability and monolayer integrity in a high throughput fashion. We actually use a device called the endothelial cell impedance assay. It's made by Applied Biophysics. And essentially, it's a plate which we seed endothelial cells on. And now we've pretty much switched from HUVECs over to pulmonary endothelial cells, microvascular endothelial cells, because we believe they're more relevant. But they take a little bit longer to grow. But anyway, the bottom line is that we seed the cells on the plate. We allow them to grow. And these plates have a mesh of electrodes at the bottom. And we're able to measure the resistance of current flow from the top to the bottom, the bottom to the top. And essentially, what happens is that if when the monolayer is intact, there is the resistance is actually high. If the monolayer is compromised, then the resistance is low, which makes sense. So we then are able to, once the monolayer is confluent, we are able to add our experimental substances of interest, and we're able to generate data which sort of looks like this. So essentially, this is a readout of resistance on the y-axis, and we're able to see over time how the resistance changes. This you can see here, the red bar is actually something that increased the resistance or decreased permeability. And we're able to look at the area under the curve to sort of get an idea of the readout. So let me go back to this slide. So essentially what we did is we used this assay, and it showed here very clearly that in a dose-dependent fashion, if you added platelets, and mind you, we did remove the plasma from these platelets because we wanted to study the platelets in isolation, that we're able to attenuate vascular permeability, or rather increase resistance of the endothelial monolayers. We also showed that this is also possible in a dose-dependent fashion to restore junctions. This is exactly the adherens junctions that we showed you before, 50,000 platelets per microliter, 100,000, 200,000 per microliter. This is all in vitro. So essentially, this is a non-leaking monolayer. And then you can ask the question, on these particular outcomes of measure, is there a storage lesion? If you store platelets between day 1 and 5 at 4 degrees, do you alter their function, their protective effect on vascular permeability? And as you know, Heather also mentioned this, that platelets can standardly be stored at room temperature for 5 days. And there's some issue of some storing at 7 days with additional bacterial testing. So with that in mind, there's also been data that's been generated that suggests that platelets, the impact of older platelets in trauma and surgery might actually be detrimental. And so with that in mind, we asked the question, are we going to see differences between day 1 and day 5 platelets on these measures of endothelial barrier function? And we see here, indeed, we do. These are fresh platelets. These are 6 donors. What's most interesting about platelets is there's considerable donor variability between platelets. We see this less so for plasma donors. We also see here that aged platelets, these these are day 5 platelets, have diminished protective effects on the endothelial monolayer. This is depicted here sort of in a summarized fashion. This is day 1 platelets versus day 5. And then we also show that the loss of protective effect in platelets is progressive over 5 days of storage. So honestly, we don't see much loss. We've looked at multiple donors between day 0 and day 3, but somehow after day 3, we start to see greater changes in protective effects. So with that in mind, we also asked the same question, and the easiest thing to actually look at is just the bottom row here. This is staining for the same junctions we talked about. So this is beta-catenin and V-cadherin. They actually bind together. This is a non-leaking monolayer, and here you see these are day 1 treated monolayers. These are day 5. You can see here that the protective effect is diminished on these particular junctions. We also find that day 5 platelets, this is an aggregation assay in vitro to pulmonary microvascular endothelial cells. You can see here that day 1, and these are non-stimulated. So essentially we're taking them straight out of the bag, spinning them down and allowing them to adhere to endothelial cells. You can see here they're stained with a black stain. So the spots you see here are the small number of platelets that are bound to the endothelial monolayer. But day 5 platelets are highly aggregative. You can see here these are non-stimulated platelets, and they seem to form a number of aggregates on this particular monolayer. So with that in mind, we also worked with Mitch Cohen's lab at San Francisco General. He's now at Denver Health. And he also showed that there was diminished effect. This is multiplate aggregation, about a 20 to 90% loss in aggregation, and velocity, which was also donor dependent. So now I'll come to the last gold standard question. So we all know that many of the assays that we look at are in vitro. And the question is do they actually translate to something in vivo? So an isolated assay, the models of hemorrhagic shock or traumatic brain injury that we use to look at leak in the lungs or the brain, actually pretty complicated models and take months to run. So we really were searching for a sort of easier to use assay where we could screen multiple substances in a higher throughput fashion for vascular leaks. So this is actually a mouse that it's an NSG mouse, so it's a non-skid mouse. It has dysfunctional T cells, B cells, and also NK cells. So we're able to look at human products in this mouse, and we treat them with the particular substance or blood product, and then we allow it to circulate for 30 minutes. Then we induce on the dorsal side of the mouse permeability using VEGF. We've looked at thrombin, we've looked at histamine, we've also looked at inflammatory cytokines like TNF-alpha to induce leak. In this particular assay, we stuck to vascular endothelial growth factor, and we're able to assess how much blue dye extravasates into the back of this mouse. That blue dye, the quantitative amount, is essentially your readout of vascular leak to albumin, because this is Evans blue dye, so it binds to albumin. So what you see here is actually a mouse that was treated with saline, and this is a mouse that was treated with platelets prior to the induction of leak. Very clearly, day 1 fresh platelets attenuate vascular leak. So this is consistent with what we had seen in vitro in multiple models. Then when you take a look, so then you ask the next question, what about the day 5 platelets? Are they also showing diminished function like they do in vitro? When you take a look at that, you can see here, this is PBS, so it's saline-treated animals. This is the skin of a mouse that was treated with fresh platelets. And indeed, the ones that are treated with day 5 platelets show diminished protection. This is depicted here quantitatively with a significant difference between day 1 and day 5 platelets. This is also shown here in terms of the donor variability that you get in platelet function. You can see here that some platelets are more protective than others. And really we went on to ask, and this is not a surprise to any of us who understand platelet biology and the storage lesion, is that it was very clear we were able to sample the blood from these animals and look at how many circulating platelets are present. We can label them with human marker antibodies, so we can isolate just the human component, and we see clearly here, it drops very rapidly. Mind you, this is a mouse model, so it doesn't mimic exactly what you see in patients, but it is indeed representing that the clearance of the day 5 platelets, or the circulation of the day 5 platelets, is diminished compared to the fresh day 1 platelets. Now, I'm going to touch on this very briefly, and Heather did an amazing job talking about the hemostatic effects. And we also, so we have a big interest in the functionality of 4C platelets. And we asked the same questions that we asked for the room temperature stored platelets on about 4C platelets. And this is, again, the ESIS assay we were describing before. This is 6 donors, and you can see here the clear bars are 4-degree stored platelets, and they're stored in plasma. And this is the 22-degree stored platelets. And you can see here very clearly that some of the donors at day 5 show enhanced protection by cold stored platelets. You can also see here that almost all of the donors, 5 out of 6 at day 7, show enhanced protection of cold stored platelets. This can possibly be attributed in terms of vascular leak to a number of factors that have been shown to play a role in endothelial leak. VEGF, for example, VEGF-C drops with cold storage, or is not increased, with cold storage. This is the case also for PDGF alpha, beta, and beta, and AB and BB. Pi1 is not increased, and CD40 ligand, it looks like it is slightly decreased at day 5, and interestingly, we didn't see big differences at day 7. So with that in mind, we asked the next question. So how do they perform in vivo with that same assay? So this was interesting to us, and it turns out that this was a split between what we saw in vitro and in vivo. And when we looked in vivo at permeability, we actually saw that day 5 and day 7 4C stored platelets showed diminished function in terms of their ability to protect against vascular permeability. This was also reflected by circulation times, which as we know are diminished with the 4C platelets. So this is an interesting area of investigation for us, and we are actually now investigating storage in other storage solutions like PAS, TPAS, and also Intersol, and we're wondering whether that might actually have an effect. It's interesting because I do not believe that this effect on vascular leak is solely due to circulation time and circulating platelets. And I'll give you this one piece of information, and the reason I'm saying that is because dried platelets, thrombosomes, barely circulate, but they're very protective on vascular leak. So I think it's going to be a really interesting area for the development of the biology in terms of what aspect of the platelets actually defines its potency and its effect in these particular areas. So this is a question one can ask: do platelets and plasma synergize? And in our very simplistic mouse assay, we show clearly that FFP has the capacity to inhibit vascular leak. And indeed, when you put the two together, they actually are synergistically inhibitory. Now, I just need to add RBCs in here now. So I haven't done that yet. So that I think is interesting in itself, and it actually asks you, you sort of wonder whether the mechanisms might be slightly different between plasma and platelets, which would make sense. And so the bottom line here, really to summarize sort of these efforts, was that we recognize that plasma and platelets are key regulators of inflammation, coagulation, and endothelial function. And they appear to synergize in some of these processes. So the question is, mechanistically, how are they doing this? And this is an ongoing effort in the lab. I wanted to leave you with one last thought of the future direction that we're working in. And we are very excited about this. And I know this field has been around forever, but I have sort of been exposed to it for multiple reasons, because it also plays out in the cell therapy arena. But one asks the question of microvesicles. So what is the role of platelet microvesicles? So it's not just microvesicles that are formed. We have exosomes which are formed. They're formed by, which are smaller. These are usually around less than 100 nanometers, and microvesicles are usually greater than 100 nanometers, can be anywhere from 1 to 2, up to 1 to 2 microns. And you really ask the question is what is the role of these entities, because they contain really very defined repertoires of DNA, RNA, microRNA, signaling proteins, other proteins, and lipids. And if you really take a look at that, they say about 50% of what nature packages into these bodies are actually reproducible between one donor and the other. So why is that? What's the function of this? So we asked the question, first of all, and this is not new, it has been shown that platelet microvesicles actually are potently hemostatic. And actually there was a whole endeavor years ago, and I don't really know, I bet you would know, what really ended that endeavor to pursue platelet microvesicles as a hemostatic agent. But clearly, when we look, we have tail snip bleeding assays. You can see here, this is and essentially it's you cut the tail off a mouse and then you put it into a warm saline bath and you allow it to bleed out. And when it stops, you actually collect that fluid and you assess for what quantity of erythrocytes are in the fluid. So very clearly, you can see here, this is a non-treated mouse. This is PBS control. If you treat them with platelets, you see diminished levels of bleeding, but also microvesicles. They clearly are hemostatic. So we asked the question, and this is actually also plasma microvesicles. So if you look at plasma microvesicles and platelet microvesicles, they actually in many ways seem to be more potent than fresh platelets and also FFP. So with that in mind, we were intrigued by this. We thought, is it possible that these could also mitigate vascular leak? And I hadn't seen any papers that were published on this area, and so we started to put them into our standard assays. And here clearly we showed that platelet and plasma microvesicles seem to have potent effects upon endothelial barrier function. And this was dose dependent. This is platelet microvesicles and this is plasma microvesicles. They seem to be basically on par in vitro. But interestingly, in vitro, we actually showed that it seems that the whole platelets and plasma seem to be more potent than they are on the tail snip bleeding assay. So perhaps indicating that in this particular area of endothelial barrier function, there might be other factors that are present in the plasma or the platelet preparations themselves. But this was the question that one would ask is, so fresh platelets, we took fresh platelets day 3, and we also took microvesicles from platelets on day 3. And we asked the question, do we attenuate vascular leak in vivo? And we see that we do. So this is essentially the graphical depiction, and this is the quantitative depiction. And we see here that this is fresh platelets, day 1. These are day 3. This is microvesicles from day 3 platelets. And this is plasma, and this is microvesicles from this donor of plasma. And in vivo, this actually looked like there was some significant difference between the microvesicles and the plasma. So with this idea, we've started to embark upon trying to understand this at a deeper level. We've started to sequence, RNA deep sequence, what's inside the platelets. We're interested in microvesicles produced by cold stored platelets as well. And you can ask the question, so why would we want to embark upon an endeavor to produce something that potentially has, as a therapeutic, that potentially has hemostatic potential? If you think about it, one of the things that comes up, and it has a lot to do with logistics and also is of intrigue to the military, as Heather would know, is the concept of being able to minimize the size, the storage needs, the portability, and of any hemostatic agent that can be administered in a far forward setting for prolonged field care, for example, of wounded warfighters, would be beneficial. So the idea here was to see if we could actually, and this could be generated really honestly from our trash. So the trash that with the unused units of platelets or plasma that are unused by the blood bank potentially could be utilized to produce these products. So these are still in its infancy, but we are embarking on this area. And then we also asked the question, if blood products have the potential to attenuate leak in general, could we envision them as treatments for other areas of injury, such as, or disease, septic shock, traumatic brain injury, AKI, dengue hemorrhagic shock? Or is it possible that there would be the ability to administer blood products in these settings? So anyway, this is the bottom line here, and we believe that a lot of this can be used as a discovery platform for novel therapies. So with that, I'll just close. I have a lot of wonderful collaborators all over, so thanks.
Thanks, that was a wonderful talk. I'm wondering if you've done any similar vascular permeability studies with either packed red cells alone or whole blood as resuscitation fluids?
No, it's exactly, excellent question. It's exactly where we want to go. So we are just starting to work with RBCs, and it's just, absolutely the next step. And whole blood, I think it would be of great intrigue. And the comparisons of whole blood to these components, for sure.
Hey, Shivani, that was great. The slide that you showed the aggregation of the slides that you showed the aggregation of the stored platelets compared to fresh. Do you think that the aggregation is because there's more vascular, there's permeability, and so it's exposing the underlayer?
I think that's a very valid question. Yeah, no, I think so, because the junctions are possibly open, so you're actually creating niches for these to adhere to. And that's actually what happens. I mean, I wish should really be studied in an intravital setup, because that's actually where you'd be able to see that. But yeah, I absolutely think that could be contributing to it. Thanks.
Hello, really fascinating talk, and really, I think, really changes the way we think about trauma resuscitation. I'd like to, without being able to directly measure vascular permeability, do you see your work sort of changing the way we approach massive transfusions in terms of ratio-based transfusion or whole blood transfusion? Or do you see the future more as we need to add vascular permeability to sort of point-of-care testing while we're doing trauma resuscitation?
That's an excellent question. So the big question is what relevance does this have clinically? And one of the things that we have been really addressing is whether or not there's a need for prolonged resuscitation even after bleeding has stopped. And that's the idea of actually utilizing blood components to mitigate long-term outcomes in trauma. So for example, if you look at the results that came out of PROPER, about 25% of the patients will die from hemorrhage, and that's within the first 2.6 hours. So they're going to die anyway. There's nothing you can do. Except if you can stop the bleeding, then you'll be able to save them. Then there's about 50% that die within 3 days. Those 3 days are mostly due to traumatic brain injury. And in that group, the traumatic brain injury group, they die from everything from cerebral edema, so brain swelling, edema, and also hemorrhage, and other really TBI-related issues. But then there's a remaining 25%, which sort of smolder over a period of like up to even 20, 30 days. And these are the guys that die from inflammatory conditions like ARDS, AKI, septic shock, all of those issues that have to do with inflammation, organ edema, organ failure. And we actually think that that's the component that you potentially could address with altering the resuscitation paradigm and potentially resuscitating patients longer. Now this is a big battle, because if you propose this to anybody in the field of transfusion, I'm sure all of you are like, some of you are going, oh my God, we would never do that to anybody. It's like dangerous. But it's a thought, because really honestly, there are aside from supportive care, anybody in critical care will know, aside from supportive care, there is not much we can do for those patients. We just kind of cross our fingers, and lung protective ventilation, but what else is there? So if we could potentially have a therapeutic that enhances the chances of these folks to survive, it might be worth it, so.
I have two very unrelated questions. One has to do with ECIS.
Yes.
So, when you do an ECIS measurement, depending on how you're modeling the data, you can measure junctional integrity. Yes. But especially with microvascular endothelial cells, you're also measuring the ability of the cell to adhere to matrix. And those are important things to separate, because one has to do with integrin activation and one has to do with VE-cadherin and PCAM and all those other things. And I don't know if you are separating those, because it makes a difference in the mechanism by which these factors may be working. So
you're 100% right. So it turns out if you coat these plates, so we don't coat them. This is all baseline,
but cells make their
without
own
laying
matrix.
down. They lay down matrix anyway. But if we coated them, so for example, with vitronectin or collagen, all of these activate integrin receptors, we have started to see differences in the
And
results,
microvascular, a large component of, as opposed to HUVEX, of the total resistance that you see has to do with cell matrix interactions, not cell-cell. So just a
Absolutely.
word of
Yeah, no,
caution
it's
there.
absolutely, I mean,
Yeah.
we let them sit in those dishes for about a day. They still can lay down
Sure.
matrix during that time. But if you already lay down matrix, you actually change the sort of, we still see the differences that we saw before, but it's clear that there is you know, there's signaling going
Yeah,
on.
and you
there's
can model it
changes
quite
in-
easily. There's software
Yeah.
to separate them.
Yeah,
The
that's
second
a
thing
great
has to
suggestion.
do with the factors that come out of platelets, or the role of platelets in thrombosis and hemostasis versus wound injury and repair. And one of them seems to be mostly platelet membranes. Like you can take platelet membranes and have hemostasis. You can have lyophilized dead platelets. But with the wound healing type of things that you're looking for, that probably has a lot to do with platelet release factors, including alpha granule contents and maybe exosomes. And I
Agreed.
was just wondering, so that's probably a lot of the reason that, so, you know, when you store platelets in the cold, their microtubular ring disappears, they round up, they lose their open canalicular system,
Right.
which is what the platelet uses to quickly release things like alpha granule proteins into the extracellular milieu. Has anybody looked at the efficiency of secretion of these things in rounded up platelets that have been stored in the cold?
I don't know, I mean, Heather, do you know if somebody's looked
I
at?
think that's really important, because a lot of those goodies are probably the things that are having your biologically important effects.
It's interesting you say that. So for secretion of microvesicles, as you know, as you're mentioning right now, exosomes, you need the contractile machinery to be able to secrete, to work, to be able to secrete
Yeah.
those. It turns out that, I was just showing this data to Heather, we actually looked at cold stored platelets, the proteome, like we sort of looked at the proteome of cold stored versus room temperature platelets. And the top, like, 15 to 20 proteins that are altered are all cytoskeletal in the cold
Sure.
stored platelets. I mean, it makes sense, right, with what you're
Yep.
saying. So I think that that's a very valid question. And, you know, if you look at some of the cytokine data that I showed, the cold stored platelets are not secreting those cytokines. It's actually...
And all the
closer
growth
to-
factors and all the little,
Yeah. Yeah, exactly. That's cool.
Thank you very much.
So we're going to take a coffee break until 10:25. For those who have posters and the judges, please do that, okay? Judge and post, and we will start right on time, yes? Okay? Thanks.